Hi everyone and welcome to this episode of Keipert Labs. In today's video we're going to look at the microscope, one of the key pieces of technology that we have used to understand more about how living things are put together from cells. So what are we going to cover in our video today? We're going to look at what do we mean by a microscope? What is this piece of technology that we use? We're going to talk about the different types of optical or light microscopes that we will be using or that we need to be familiar with. And we're also going to look at and unpack each of the parts of the microscope. So how is it put together and what do each of those parts do? So firstly, what is a microscope? It's essentially an instrument, a piece of equipment that we use to view very small objects. Okay, so they might be as small as a single cell. It might be a piece of like a piece of a plant. It might be a very fine, very tiny piece of mineral or rock. It doesn't have to be only to examine living things. Now, we used it to examine living things at the level of the cell, but it's purely just a technique that we can use to make small objects look bigger. The way that it does that is through a combination of lenses, so different kind of bits of ground glass or kind of perspex or whatever that might be, that when we put them in combination together, that help to turn a very small object into what appears to be a very large kind of image of that. Now, one thing that it does, I don't want you to agonize over this diagram too much, but it's just saying that there's a combination of two lenses that we use that then will turn a little object into a virtual or pretend larger image that we see. But one little thing that I want you to look at here is that this object here, this little arrow is pointing one way, that the image that your eye will pick up is actually upside down relative to that. Okay, and telescopes work much the same way. The microscope is kind of the same sort of principle that works in a typical simple telescope, but just built in a different way to look at small things rather than looking up at the sky. And so taking this idea of looking at very small things, making them look much larger, is an idea we call magnification. And so depending on what each lens will do, we talk about different number of times magnification. So 40 times magnification means that something that's 1 millimeter in size in the image here is actually 40 times larger than it is in real life. Likewise, this is 100 times larger, 200, 400 times larger. You can see it looks like you're zooming in just because of the different combinations of lenses that make the image look bigger. Okay, so this idea of magnification. So we have three main different types of optical, that is light-based microscopes that we use in common use. So the first one is what we call a monocular microscope. So mono is one, and ocular, it refers to eye. So monocular is one eyepiece, or you look through it with one eye, you can see here. So then it kind of makes sense that if I talk about a binocular microscope, you can see it has two eyepieces. You look into it with both eyes at the same time. And so, because bi is two. And so one eyepiece, two eyepieces, but you're still looking through one lens down kind of at the bottom. So you get a very 2D or kind of flat image. You can only look at very thin things and see them in focus at the same time. The third type of microscope is called a stereo microscope, also often known as a dissecting microscope. So that this works a little differently because you have two eyepieces, but you actually have a combination of two lenses that are focused on the image at the same time. What it does is that it actually allows you to view things in 3D in the same way that you have two eyes that allow you to see objects in 3D, because each eye is seeing the object slightly differently and your brain can combine the images together to allow you to perceive or understand depth. Okay, and that's one reason why if a person loses one eye, they often have difficulty in being able to do that they're not allowed to drive and things like that, because they can't perceive distance or depth the same way we can. So a stereo microscope allows you to view objects up close as though you were seeing them in real life, rather than just a flat kind of image. The reason we call them dissecting microscopes is that if we are trying to dissect and we're trying to look at fine details and actually do things at the small scale, but be able to see them in real time, we need to use this kind of microscope. The monocular and binocular microscopes are all to just view samples that have already been prepared. Okay, so you prepare them, you put them there to look at them, but you can't actually work with them at the same time. Stereo microscopes you can. Okay, so now here we have a kind of a side view of a binocular microscope. We're going to go through each of the different parts of this, and you're going to have this image that then you're going to label. Okay, so firstly we have the base, the wide kind of stable bit that allows the microscope to sit flat and secure on the bench. Inside the base we have a light source called a lamp. So basically a light globe that points up so that it shines through the sample in order to then the light can travel up through the lenses and into your eye. So a stereo microscope works a little bit differently as far as that it might have a light pointing down because you're looking through the sample as it is. You're not relying so much on light that shines directly up into your eye. But for this one, the lamp points up and through the sample. To control the amount of light that gets through is what we call the iris diaphragm. So this functions like the iris in your eye. It can open and close down to allow more or less light to travel through, depending on how much brightness that you need or how easy it is to see the sample you're looking at. The sample is placed on the stage, this flat bit here that's able to be moved up or down, and it's also some microscopes it can also be moved backwards and forwards or kind of moved in other dimensions as well. Okay, in order to hold your sample still on the stage, we have what we call stage or slide clips. Okay, so the microscope slide is a little piece of glass that we put our sample on, and we need to make sure that it's going to stay still, but we don't need to constantly hold it, that we can be hands-free or use our hands for other things when we're working the microscope. So the stage clips hold it still. And so in order to be able to see the image clearly, we need to be able to focus on it. And so what we have is we've got two different focusing kind of knobs on any given microscope. We have what's called coarse focus and fine focus. So coarse focus is what we use. It makes big movements to move the stage up or down to allow us to focus on the image. Okay, so it's lots of big changes. Okay, whereas the fine focus, as we turn it, it makes very small gradual changes. So as we're really close to being in focus, but we need to just make tiny adjustments, we use the fine focus and it tweaks it just a little bit. And especially if we are looking at things under high magnification, we only ever want to change it a tiny bit, because otherwise we're just going to go from one blurry image to another blurry image, and we're never going to see anything clearly. So coarse and fine focus. So you can see on this microscope, in a similar way to many of the ones we would use in the lab, that they're both kind of connected to the same dial. The fine focus is kind of the inner kind of bit, and the coarse focus is kind of the outer larger bit. That may be different depending on the microscope. We then have the arm, this kind of section that connects the base all the way up to where you are looking through. We then have what we call objective lenses. So these are the lenses that actually point at the sample and that they spin around on this turret. So we have in this one we have three different objective lenses. We call them different power, different magnification. And you can rotate them around depending on which one that you need to look at the sample you're looking at. They spin on the turret so you can see, maybe 4 times, 10 times, or 40 times, for example, so that to look at different levels of detail. And up at the top, then we have a second lens. It's called an ocular lens or an eyepiece. That is the one you are looking directly through. So you're placing your eyes up on that one so that you're looking through that, and the light travels through there and into your eye so you can see the detail clearly. Okay, so we're looking at this idea that we have two lenses essentially in our microscope. We've got one here and one here that together in a compound way that then they are giving us the magnification that we need. So typically an eyepiece has a 10 times magnification. That is, the image is 10 times larger purely because of what the eyepiece does, and then the objective lens will be an additional magnification, 4, 10 or 40 times. Okay, so we times those together to give us the magnification that we're looking at. Okay, so we've looked at what is a microscope, in terms of being an instrument that we use to look at very small things to make them look larger, magnify them. We look at the 3 different types of optical or light microscopes, monocular, binocular, and stereo. And we looked at the different parts of the microscope that work together to help us to be able to view samples, to be able to magnify them. All right, thanks very much for watching. Bye for now.